Good day, everyone. You are welcome to another schedule of Practical Pharmacognosy 1, that is PCG 212. This schedule is a continuation of microscopy of crude vegetable drugs with emphasis on transverse sectioning and histochemical techniques of leaf sample. This slide cast will follow the outline as listed. The topic will focus on the following objectives: preparation of leaf transverse section, microscopic examination of the section, preparation of leaf epidermal surface and its examination microscopically, preparation of powdered leaf sample for examination under the microscope, histochemical techniques for microscopy of leaf drugs. At the end of this schedule, students will be able to prepare transverse sections of leaf drugs for examination under the microscope. You will know how to prepare slides for the microscopic examination of both whole and powdered leaf drugs, as well as mount, visualize, and correctly identify the diagnostic microscopic characters in a leaf drug. Transverse sectioning of a leaf drug is part of qualitative microscopy to understand the arrangements of various tissues in the sample. Surface view of the leaf epidermis, on the other hand, helps mainly in the examination of stomata and attached trichomes on the abaxial and adaxial surfaces. That's talking about the appearance, the types, the distribution. Examination of the leaf epidermis in surface view also makes quantitative microscopy possible to determine the stomata number, stomata index, vein islet number, vein termination number, as well as the palisade ratio. Histochemical techniques are useful for the visualization of biological structures using specific dyes, both on powdered and whole drugs. These are the materials and apparatus required in this schedule. For the purpose of this slide cast, we are going to focus on two leaf drugs: Azadirachta indica, commonly called neem, and Cassia Senna, commonly known as Senna. A stoma is microscopically small, but a very important structure on the leaf surface. It consists of a pore that is surrounded by a pair of specialized cells called guard cells, bringing about gaseous exchange and transpiration in the plant. The number of stomata, the size, and location vary across plant species and can be influenced by a variety of environmental factors, which makes them highly diagnostic in any leaf drug. But stomata are not easy to see on the leaf surface, even under the microscope. Therefore, an indirect approach has to be used by first making the leaf transparent, either by scraping, peeling, clearing, or cuticular method. For example, the epidermis of Talinum can be easily removed by peeling. For the scraping method, the epidermis of most leaves can be removed by scraping, e.g. Azadirachta indica, Jatropha, just to mention a few, except for those with extra surface coverings like numerous hair, waxy cuticle. In that case, extra patience is required. Fresh leaves are required for both the scraping and the peeling method. But for the clearing method, either fresh or dry leaves can be used. The peeling method is easy and fast. It's simply done by partially breaking the surface of the leaf and slowly pulling one part of the leaf towards you until a transparent layer of the epidermis detaches from the leaf surface, but make sure to avoid the midrib area. The epidermis of the leaf can be scraped with the aid of a razor blade held perpendicular to the leaf surface. In this case, you scrape gently, adding solvent intermittently as required until you have enough clear epidermis, but make sure to avoid the midrib area. For the clearing method, a clearing reagent is required. Warming gently, but make sure to cover your sample with a cover slip and keep your slide constantly irrigated until you get a clear epidermis. For the transverse sectioning of a leaf sample, two methods can be used. First, by cutting out a rectangular piece from the widest part of the leaf sample, including the midrib, and embedding it within a cork. This helps to hold it in place. It makes cutting of very thin slices easy. In the absence of this, another method can be used. Here you take out one leaf and roll it. Cut through the roll to obtain a very thin, almost transparent slice. Whichever method you choose to use, make sure to cut several thin slices. Then transfer the sections into a hollow glass with water to separate them, and with the aid of a hand lens, select a very good section. Place the section on a glass slide using either a small brush or a pair of tweezers. Gently place a cover slip on the section and irrigate with dilute glycerol. Remove excess liquid using absorbent paper and examine the slide under a microscope. Histochemical examination of leaf samples helps to quickly detect the chemical nature of the leaf drug using very minute quantity of plant material and microliters of reagents. The reagents impact specific color depending on the nature of cell inclusions, namely cellulose and lignin in tissues, oil cells, mucilage, and others. Some histochemical reagents commonly used in our laboratory have been listed below with their specific applications. Now, the purpose of several listings in this slidecast is for reference in case you decide to print out the PowerPoint slides. Adding mountants to the prepared specimen before viewing under the microscope is not for the fun of it. The mounting medium binds the specimen, slide, and cover slip together with a clear, durable film. Of course, you have to make sure that your slide is not cloudy before viewing under the microscope. It also helps for the image formation. Glycerol has a refractive index very close to that of the tissue mounted, which helps the specimen's rendition. Water and dilute glycerol are the most common universal mounting medium. However, glycerol can stay longer for a few months, keeping the slide flat, unlike water mounts. The addition of glycerol also helps to prevent the crystallization of chlorohydrates where needed. The images A and B were placed here for you to appreciate the stomata type in a very common leaf drug as a Directa indica, family Meliaceae, commonly known as neem. The scraping of the leaf epidermis revealed an homocytic type of stomata. Interestingly, the stomata appeared only on the lower epidermis. First, you focus on low power, and once you can get a very sharp image before moving to high power to draw. An homocytic type of stomata because the subsidiary cells that surround the guard cells cannot be distinguished from the other epidermal cells. Here, the subsidiary cells are 5 in number, But please note that in an homocytic type of stomata, the subsidiary cells are not fixed. The number of subsidiary cells are not fixed. For example, digitalis also has an homocytic type of stomata. So you don't expect that it's going to have 5 subsidiary cells. Whereas in diacitic type of stomata, like can be seen in Kigelia africana, and parallel parasitic type of stomata, that's the parallel celled type of stomata, like can be seen in Cassia Senna, the number of subsidiary cells are fixed, just 2. Also note, the magnification here is just 1. But if you were to draw in your manual, you are expected to write the eyepiece, the objective lens, and your personal magnification, say 2. This is very important. Otherwise, you are not going to get your complete mark. This image will help you to appreciate the sectional view of Senna leaf cut transversely, and it will help you to understand the arrangement of leaf tissues. Images 3A and 3B show cleared Senna leaf in surface view. In 3A, we can appreciate the parasitic type of stomata found on both the lower and the upper epidermal surface of Senna leaf. We can also see that the epidermal cells have straight anticlinal walls. The type of trichome is clearly seen here to be a covering unicellular warty type of trichome. It will be important to mention here that safranin dye stains both lignified and unlignified elements red. However, safranin displays exceptional cellular details. Please note that when a trichome is lost, a scar remains called cicatrix. So do not confuse this to be actinocytic type of stomata. Image 4 shows cleared Senna leaf powder. In 4A, we can see a bundle of fibers with calcium oxalate prism sheets. In 4B, we can clearly see a unicellular covering warty trichome, though it's broken. It will be important to mention here that stomata is not the only diagnostic character of a leaf drug. Trichomes have protective and sometimes secretory functions. They show great variation across plant species structurally. Therefore, they are highly diagnostic. Likewise, calcium oxalate crystals vary greatly in shape and distribution across plant species. They are also diagnostic. At the end of every laboratory report, you are expected to have a section for comments or discussion. This is just to discuss your observations, results, and nothing more. I had to put this here because I know you are familiar with this kind of image, like you always see in your Trees and Evans and other reference books. What this means is just bringing all the diagrams together. It's a complete diagram that shows the microscopic features of Senna leaf, both the sectional view, the surface view, and the powder microscopy. For example, Figure 1, Figure 2, and Figure 3 are in surface view, as you can see, whereas Figure 5 is a sectional view. But figure 3, figure 9 are from the powder. As you can see, they are isolated. These are clusters of calcium oxalates, and these are trichomes. These trichomes are not attached to the epidermal cell. That shows that it's from the powder. Here also is from the powder because you can see it's in fragments. Though the trichome here is attached to the epidermal cell, but you can see that it's in fragment. Fragment means it's in powder form. Likewise, 8 is a fragment of the xylem vessel, which is pitted. So in summary, this is the report of microscopy of Senna leaf. There are some precautions that you have to note before starting with this experiment. Number 1, chlorohydrate solution is corrosive and irritant to the eyes. So you have to be careful how you use it. Do not allow it to come in contact with the objective lens of the microscope or the stage because it could corrode the cementing layer. Also, it sedates, so do not inhale. Fluoroglucinol is explosive. Be careful with the naked flame. Calcium oxalate crystals and starch disappears after addition of HCl. So, before you add HCl, you have to examine the sample for the calcium oxalate crystal and starch granules. Likewise, nitric oxide must be used under the fume hood. In conclusion, this practical schedule is useful as students can now understand the arrangement of leaf tissues, distinguish between the types of stomata and other diagnostic characters in leaf drugs, as well as appreciate the relevance of histochemical techniques in the examination of leaf drugs. I recommend these textbooks and references which I used in the preparation of this slidecast. Thank you for your attention and stay safe.